Good afternoon. I'm going to give you a quick overview of the NUMAR model, Numerical Understanding of Marsh Accretion and Resilience. This is a unit model, and this is an introduction to help you understand more of the concepts related to the model simulations that you're going to run later in this portion of the workshop. So let me go right into what we one of the things that we really emphasized when we were building the model NUMAR 2.0, and this comes from a basic soil cohort model to predict soil accretion over a 100-year time frame. And what we wanted to do was to expand the ability of the model to simulate very different environments within the delta, and the focus was on a delta basin that's where the wetlands are stable and growing and developing versus the deteriorating, the unstable coastal basin. In delta cycle, you refer to this as the active delta cycle and the inactive delta cycle. And can we actually program this model and develop it so that it can actually accommodate both of those aspects of the delta cycle, the building cycle and the deteriorating cycle. And in Chafly, you can see this white line is the shoreline in 1932. The blue is a shoreline based on wetland to water ratios of 50%. In 2010, and you can see that in the Chafly, these shorelines have been stable, that there has been no migration, whereas in the Terrebonne, this migration is nearly 10 miles during this time frame. So again, that's the idea. So we established sites. This is part of the Delta X project in these two basins to compare, again, in this Delta cycle concept, what we refer to as the active and the inactive. Another way to think of it is a sediment rich and sediment poor. So again, can we develop a model that allows us to capture all the parameters accurately to look at soil formation? Because we know that the ability of wetlands to sustain or to remain in these landscapes is based on the ability of building elevation, what Don Cahoon refers to as elevation capital, and building that elevation relative to sea level rise and subsidence. And so that's why we wanted to check this model again in those two very different types of environmental settings, but also along a salinity gradient. And so thus the experimental design, and I'll talk a lot about the generality of the model to pick up accretion in these different settings. And just to give you an idea how different these settings are, just look at bulk density. Again, the first three bars are the salinity gradient from fresh to saline in the active delta zone. And the same, these red colors are in the inactive, again, from left to right, going from fresh to saline. And the highest bulk density in the active, obviously, is in the deposition in the Wax Lake Delta region. That's your high bulk density. Whereas in your inactive delta, the freshwater sites are floating marshes with very little bulk density. And actually, the bulk density increases as you go down to the salinity gradient. And if you look at inorganic matter loading, again, the highest inorganic deposition is in the fresh zone of the active, but it's actually in the salt zone in the inactive. And so, again, this is what we wanted to be able to develop this soil formation model, which is, again, based on a model format that actually goes all the way back to the grasslands LTER back in the 70s. And see if we can build it to accommodate those different settings. Now, I want to make sure our terminology here, it can get really confusing, and I'm going to try to be as clear as I can on this of what we're actually modeling. We're modeling the top 100 years of soil formation. And that's that surface accretion, if you will, that elevation that the soil is building over that time frame. Thus, the organic matter becomes a real important part of that elevation gain. Now there's shallow subsidence, there's expansion and contraction of the layers beneath this zone, and that also contributes to surface elevation. So you do not get the absolute surface elevation of a platform with this model. But what it does is it gives you that accretion component, again, over 100-year time scale. When you take that and account for subsidence, then you get an idea of what the net elevation change is of the surface. So I just want to be really clear on that because it's very important part of this exercise. Feldspar, I want to be really clear. A lot of times it's referred to as vertical accretion. Again, the model, I'm going to use vertical accretion quite a bit. That's what the model generates, the unit model, again, on a 100-year time scale. We use marker horizons to give us surface deposition. And so there are two major factors that build the soil, and that is what is deposited on the surface and what does the below ground production of roots generate. I know a lot of people say, well, the marker horizon picks up the below ground production. It doesn't. You're only looking at, just a couple of centimeters at most of deposition. And then you got root production that goes all the way down to 30 or 40 centimeters. So that's, again, I want to be really clear that we're going to use the marker horizon and we're going to use that as to parameterize the model to give us the annual deposition of inorganic and organic matter. That's very important. And so you lay down the idea, if you're not familiar with marker horizons, you lay down a white feldspar clay material. You can see it here in this photograph. It lays down right on the surface, and then you come back periodically and measure what is deposited above that white material. And you can see in the sediment rich down here in the bottom, you see quite a bit of material deposited on top of the white clay, and we can go back and measure that at different time intervals, and you can see in a sediment poor basin how much less material is deposited on the surface. So thus the marker horizon is surface deposition, and that's a very important parameter that goes into these unit models. You can see here what we do in the field is we go out and use this device where you actually put a tube down in the soil and then inject liquid nitrogen and freeze the soil, and you come up with what we refer to as these popsicles. This soil is frozen onto this tube and pulled out. Once it's frozen, you pull the tube out and the soil comes with it and gives you, frozen, if you will, in time, this structure of where is the marker horizon and above which is the deposition. So you go back and you deploy the marker. We did in fall in 2019, and we go back 1 year later, measure the deposition above the marker, go back another 6 months, measure again, go back another 6 months, measure again. And what you end up with is you end up with a series of measurements at these different times since the original marker was laid down, and that gives you, again, to see this is surface accretion. You'll see that quite a bit. I want us to focus that this is deposition. This is the deposition in centimeters per year of material on the surface of the marsh. Now, again, that's the surface deposition that builds soil formation. There's also the below ground processes, and the way that we capture both of these is what's known as a soil cohort model. And basically what we do is each year we lay down, it's a 1 year time step, each year we lay down a layer of soil, and then we track what happens to that layer each year over 100-year time frame. And so the layer that's laid down on top, of course, has root material, inorganic matter, and has this material deposited on top of it. It also has organic matter. And so you have inorganic matter and you have organic matter. And this inorganic matter, again, that's the sediment inorganic matter that's laid down at the top, that's deposited on the top. And we track that over 100 years. And of course, it is conservative. What you lay down in 1 year stays in that cohort throughout the time of the simulation. The organic matter, of course, has a fate dependent on what new production occurs in a year versus what decomposition occurs in a year. One is it adds new organic matter each year, and decomposition removes organic matter each year. And those rates, relative balance of those rates, that net is what is the organic matter that contributes to vertical accretion. And again, we track these year to year, and the sum of which is a formation of the soil. That's again the composition of both organic and inorganic matter. So we start with this surface deposition. And again, feldspar gives us both the organic and the inorganic. Now, how do we do that? We just take a layer of the material that's been deposited and ash it, heat it at about 500 degrees centigrade. And the material that burns off, or the loss of weight, if you will, due to heating it at that level, is the organic matter that burns off. So you lose that weight. The weight that remains after you burn it at that temperature is the inorganic matter. It's a very simple definition. And so that's how we get the inorganic and organic matter deposited on the surface. And then again, that's the surface component of the unit model. Then you have to add what the roots are contributing. And that is a below ground production. And we have to account for the amount of root material with depth. We use this distribution model, which is a negative exponential, and that gives us the amount of root material at the very surface and the incremental decrease in root matter with depth. And we use that together with the turnover rate to give us an idea of root production at each depth. And that becomes a very important part of the equation. We use this factor called E, which is basically the rate at which root mass decreases with depth. And that's, we spent a lot of time in the field measuring what this attenuation coefficient, if you will, is for root mass with depth, and that's been part of our field experiments. And you'll see those values established in the as a model parameter. Now, your organic matter, you have to account for what you lose, and that's the decomposition. And this gets into a very important distinction, and that is what we call labile and refractory. Again, this is probably going to be where we're going to spend a lot of our next research efforts, because this is a very important component of these models. The model is very sensitive to this coefficient. We've been using lignin. Now we're starting to incorporate all of the other inorganic, the cellulose, hemicellulose components of the refractory. And what we have been doing in the last couple of years is really capturing this necromass component. A lot of people just throw this away. They just wash it out and don't capture and measure it. And we've found that, you know, you really have to know what this necromass. We just can't look at the dynamics of live roots. That's just been the last, you know, what we focus on in ecology, and has very important ecological implications. But if you're going to build these models, it's the total mass balance you got to account for. And so again, this is where we spend a lot of time. And again, this decay is what we're going to do a lot in the future. So I thought I'd just take you real quick, you know, to capture this cohort and what you're simulating when you get to the models and what you're working on. Again, you've got a single cohort. And each year you add material that's deposited on the surface, and you add the material that's produced from the roots. That's your mass balance. That's your inputs. And then you have to subtract from that what you lose by decomposition. And the sum of all those is what's left in this box, and that's what builds your cohort. And then what you basically do, well, again, here's the below ground biomass. The amount of root production, again, is based on this attenuation of root mass times the turnover rate, and that's what gives you the input of below ground biomass. And here's how we define the E coefficient the attenuation rate. We measured root mass with depth and probably more detail than what you really need. But again, this is where the research in the field helps us parameterize these models. Now, the other part again is the decomposition and separating out the inorganic and the organic. And we do that again by heating material, getting the amount that's lost on ignition. Again, it's lost when you heat it to 500 degrees. And that gives you your inorganic and organic material. And then of that organic material, you can send it to a lab and get the lignin composition, and that gives us one part of the refractory material. What we're going to do in the future is we're going to add cellulose to this. We think that that's a missing component that's really important in these models. And what you have left, taking the mass balance approach, if I know how much inorganic I have and total organic matter, of that organic, I know how much lignin I have, then the part that's left over is the labile. And so we've been doing it by difference. And again, I think what's the error in this is you put more weight on labile because we haven't accounted for cellulose, but that's our next research adventures. So that's how you do the mass balance. Those are important components. That's why you'll see those reference to those parameters in the model. Then you add you've got your one cohort you started with, and now the second year in this simulation, you're going to add a second cohort on top of that. And again, you do that by how much is deposited on the surface and how much is the below ground production. And that gets laid down on top of the cohort that you built the year before. Go through the same exercise. And then you do it the third year. So the third year of the simulation, third time step, 1 year per time step, then you add another cohort. And as you can see now, you're just building these cohorts on top of one another. And that's what's giving you the elevation change that we refer to as surface accretion over 100 years. And you probably over 100 years end up with several of these, stack of these cohorts on top of one another. Now, one parameter I want to emphasize real quick is called the self-packing density. And this is really critical because the way we actually calculate accretion and elevation is the mass balance. We take the mass balance of inorganic matter and we take this self-packing density of that inorganic matter in milligrams per cubic centimeter, and that's that BI coefficient you're going to hear a lot about, and then that gives us cubic centimeters of material at a centimeter square area, which basically ends up telling us what the increment of centimeter height that we've added due to the inorganic mass balance that's in that cohort. Do the same with the organic matter. And with a self-packing value in milligrams per cubic centimeter, if we know the mass balance of organic matter in that cohort, then that gives us the cubic centimeters at a 1 centimeter surface area, 1 centimeter square surface area, and thus that gives us the depth of that cohort due to the organic matter contribution. And so this is how we generate the elevation change. Again, is based on this mass balance. So if you produce too much organic matter or if you decompose too much organic matter, that organic matter mass balance is off. And if it's off, then your elevation is off. Or if you're using the wrong self-packing density, you could have the mass balance correct, but you don't have the right self-packing density. This is where I think some of the great improvements in the model, and this is the real challenge of being able to build generality in these models, being able to account for these parameters. Pradipta is going to go through quite a bit of that. I'm just showing you some of the different values. The BO value ranges from 0.05 to 0.09. The inorganic self-packing density, though, is pretty much spot on 1.99 across all our sites. And so, again, this is something that we've been working on in improving these models. The last point here is root turnover rate. These values are all over. The methodology is not very specific. There's a huge coefficient of variation in these estimates, but this is another extremely sensitive parameter to the elevations that you generate, again, because it's the rate at which organic matter is produced. And again, Pradipta will take you through some of the parameters or some of the ways to parameterize turnover rate among the sites. So this is what you end up with, again, is a composite, and it's, again, mass balance, and the parameters that you're dealing with characterize the inputs, characterize the outputs, and then the quality of the material, and then the self-packing parameter. And you get these three outputs. You get the long-term accretion rate, again, long-term, 100 years. Let's be very careful here. You get an organic matter density in grams per cubic centimeter, and that allows you then to calculate the carbon sequestration rate. So not only are we interested in knowing the elevation change across sites and what factors control those elevation changes, but what the ecosystem service and carbon sequestration in this process. Now you can take this unit model and apply it across a landscape. I'm not going to spend a lot of time. You'll hear other people talk about this. You can have a different cells, and then you populate those cells with the parameters of the unit model, and therefore you can run the unit model inside each of those cells. And as such, if you have an accretion rate inside each pixel or each cell, if you know the parameters within that cell for the unit model, then you get a landscape estimate of accretion rates for wetlands in that region. And the Delta X program, the focus has been to use satellites, to use the remote sensing capabilities of NASA to parameterize the model. That's been the whole one of the major objectives of the Delta X program. So we can have a high resolution map with these cells. We populate it with above ground biomass based on a map that NASA generates. We use field measurements of the ratio of above ground to below ground. That helps us parameterize the roots. And then we have the E value that is a lookup table for the cells and that's usually by different salinity gradients, and then that gives us the RO. RO is a critical parameter because that sets up the amount of mass that we're going to produce each year. That's an input to the model. You can also take your feldspar measurements across the landscape, put that in a lookup table that gives you the surface deposition, populate your cells in a landscape, and that's what you're seeing here on the left is an image of the different cells in a landscape. In the middle, that's a map of vegetation. So you parameterize the vegetation in each of the cells based on a map such as this. And you take the E value, take the values of surface deposition from our feldspar, populate cells based on those measurements, and then you get a map that gives you accretion rates. And here's your lookup table. So again, you have each cell, each pixel here on the left, and then you just populate those parameters in each cell based on whatever information you have available to you that gives you those landscape definitions. You'll hear more about this during the talk. Now you can get really sophisticated, take another model, say that gives you inorganic deposition, such as this a DELT 3D model or some other NUGA model, inorganic mass accumulation on a landscape. All of a sudden that becomes your input variable for each of the cells that you want to run the unit model. And so you have a high resolution map, you take these inorganic input values, and from that, we have to use a ratio to give us the organic, and then you have to make some assumptions in the ratio of lignin and cellulose related to labile organic matter. A lot of assumptions goes in the ratios. Once you get one variable, you use these ratios to generate other input variables. But again, you end up with an input table, a lookup table, and you populate it with different techniques. These are the variables that you need for the unit model across each of the columns, and you use different methods to populate those variables in each of the cells, and that gives you the ability to do these landscape models. And so it's a lot of diversity, a lot of ways to do this. Be creative, and again, it all goes back to the unit model. It's the unit model that builds the fundamentals of how soil accretion occurs. So I'm going to stop there and let the modelers turn it over, and then have a good day.
All right, so we're going to do a few more slides to talk more about the details of the model and then have the 2 tutorials.
Hi, I'm Pradipta Biswas. I'm a graduate student at LSU. So you can consider me as a kid. So if I make any mistake, you can raise your hand and stop me there. No problem. So Dr. Twilley has already talked about the model simulations and all other stuffs. I will just give a quick recap for our evolution of our model. Oh, it is not updating here. Somehow I have to look at here. So at first, they developed a model. So in 1999, Dr. Twilley and Dr. Chen invented this NUMAR model that was nutrient mangrove model. It was basically applicable for the mangrove environments. It was actually adapted from Morrison-Bowden's semi-deck model, and that Morrison-Bowden's semi-deck model was actually from grassland model, inspired from. It's a cycle of inspiration. So then Andy developed the NUMAR 1.0. I'm acknowledging it in NUMAR 1.0 because we have made some changes there. And when I came here in August 2022, I started to work on NUMAR 2.0. So we removed time inconsistency, then removed added course root contributions, and added necromass contributions, cellulose, etc. And from all 3 models, combining all of these improvements, we are introducing NUMAR 2.0, and both of the versions are in internally review right now. So Dr. Twilley already has talked about the workflow of this model. So you have to calculate the cell packing density of organic matter and inorganic matter. And you have to calculate the root biomass at the surface and its attenuation rates, because root is the only thing that can contribute and change the organic matter concentration inside the soil other than the surface deposition. And then we have to calculate the root turnover rate. Root turnover is basically introducing some dead roots in the system. So these were the variables we used for our model. So I will talk about the source of variables first. Then we'll go and talk about a little bit about the estimation or calibration of the parameters. So the organic matter loading rate on the phosphor and inorganic matter loading rate both are from Casaway et al. 2024. And all other parameters like lignin content in the dry mass, then ash content in the biomass, root distribution parameters, etc., lignin content in the fine roots, all are those taken from Andy's thesis. Cellulose decomposition rate and lignin decomposition rate are from Means et al. 1985. And the cellulose content in the biomass, marsh organ, from Wilson, 1985. So we had to do some kind of calibration in the variables. We had to calibrate the cell packing density of organic matter, cell packing density of inorganic matter, root distribution parameter, that is basically root attenuation rate, live root biomass, and fine root turnover rate. So how did we calibrate the parameter? So at first I will talk about the root biomass at the surface and root attenuation rate. So from field measurement, we had root biomass at the surface and root biomass at the root biomass for the whole course up to top 50 centimeters of soil course. So we have different intervals and different biomass, so we can convert those things as a point data. And then we have the equation for root biomass at the surface. And we are considering that all the root biomass are attenuating exponentially with the depth. And we can define an objective function where ROB is the observed data point, and our PRE is the our prediction, which is basically from the iteration we are considering. And after the iteration, it will give you the best fitting root biomass at the surface and root attenuation rate. Similarly, for calibration of self-packing density of organic matter, inorganic matter, we are considering a constrained optimization method developed by Levenberg Markdoth. So similarly, you can define objective function that is directly objective function between the prediction and the observation. And OM and IM is not the percentage, it's fractions, so gram per gram. And BD observed is bulk density from the field measurements for each cohort, 10 centimeter cohorts. And the prediction, BDPRE is the prediction from the iteration. So whenever it optimizes the data, it will give you the self-packing, best fitting self-packing density of organic and inorganic matter in gram per cubic centimeter. So we had to do some root turnover calibration because in literature, most of the values are scattered. Like some claim the root turnover rate can be 0.04 to 0.44, and some claim that it should be more than 1, then up to 3.22. So we are not going into there. We just set a range like 0.04 to 3.22, the lowest and highest, and did some Monte Carlo simulations, and then calculated the top 50 centimeters organic matter percentage, and then saw which index for root turnover rate actually matches it. Very near to the field measurement, field's organic matter percentage. So are those calibration really fits our experimental values? So we calibrate the BO and VI, self-packing density of organic matter, inorganic matter. All the red dots here, all the red lines are from observed value, Castaneda, MOA, and Solohin, and the blue lines are our model produced value. The calibrated, using our calibrated BO and BI value. And root biomass at the surface, you can see most of the sites has a good fit of root biomass at the surface with a good R-square value. And for the root turnover rate, we can actually predict root turnover rate. Kind of estimation probably. It's not the best fit, but we can estimate. So now we have self-packing density, we have root turnover, we have root biomass at the surface, so we can populate all the files using Andy's lookup table and run our code. Alex, would you like to lead this landscape portion?
So we're going to do the first example. This is the unit model.
Can show both. Okay, cool.
Okay, we're going to do it in Google Colab, but it will work on your computer if you have installed the NUMAR YAML file in Python. So from the GitHub page, why don't you pull up the GitHub?
You can just see the instruction manual and go to the GitHub page. Okay, I'm zooming my screen.
That's good.
Yeah. So you have to go inside the tutorials. then the NUMAR model.
We've also included the two relevant references here, the one from 1999 and the one that just came out in 2024 for your reference.
So if you just scroll down, you will see tutorial 6 build around NUMAR unit model. So you have to click open in Colab. Okay, you have to run each of the cells. So just hit on the triangular sections. It will ask to give you permission. You have to hit run anyway.
And then the next two are currently commented out, but if you're running in Google Colab, you'll need to uncomment them.
Yeah. Uncommenting means you just have to remove the pound sign. So I'm removing all the pound sign. And running it. So it is cloning the DeltaX workshop repository. It will take some time.
You have to pause a moment so Mark can catch up. So we're doing the first one, 6A. Yeah.
So after that, you have to run. I mean, keep just running the cells. It will give you the output. So it run on the next cell. It will construct its path. Yeah. So if you want to change anything, like if you want to change the input file, you have to change here. Site parameters at CSV.
What is currently set to run?
So it is actually locating its main folder where the load code is located. And then inside the input parameter folder, the site parameter CSV is located.
Which sites are we running right now?
Oh,
Or how did you determine the input parameters?
so we are just running for active brackish. That is Screams 399. Active Salin, that is Screams 322 site. And inactive brackish and inactive Salin. I can pull off the slide, I mean, the CSV file.
And so the parameters for this were determined from field measurements.
Yeah.
Correct.
All of the parameters are from
Yeah.
field measurements.
Yeah, so each of the CRIM sites he mentioned are from the DeltaX field campaign. So all of the parameters for the model were measured in the field and are being input into this version of the model.
Yeah, you can actually download this file and make modifications. So the file looks kind of like this. I'm zooming it.
Yeah, so at the end of Robert's presentation, he showed this table. So these are the actual values that are used for the model. And I don't know if anyone besides you have memorized what all of these are, but there is a table in the slides so you can read about each parameter.
Yeah. So we'll keep running the cells. So if we run this cell, it will load your data frame. And then
Hold on one
have to
second.
scroll
Hold on one
down.
second. Did you uncomment?
You have to scroll down and
In Colab,
run
you have to uncomment.
this cell. It will start to produce your graph and files.
Yeah, so that cell is the model, is the NUMAR model itself.
Yeah. You can just look at it and see what formulation we are using for our model and other stuffs.
And this is a Delta X product that is in review, so it will be published once it goes through full review with ORNL DAC. And
Yeah.
that will include the model and these example sites.
And then you have to run the final cell. If you run this cell, it will give you the 100-year summary for each of the sites. This is our 100-year summary. And on the left-hand bar, you can go to the output files here. You can see after the input parameters, there will be output files. If you click there, it will create two subsections like individual sites. So you can download each of the CSV file from here.
Can you explain what each of those, what you've printed out there? All of
Which
those
one?
parameters, or not all of them, but what those parameters are?
Which one? Oh,
yeah,
this one. Okay, so it actually predicts the total accretion for 100 years, also the mean accretion for mean accretion in centimeter per year. In the accreted material, what is the mean organic matter percentage and mean organic matter density, bulk density, and carbon sequestration, organic carbon density. So, and also the necromass in the soil. And there are different kind of necromass. So we actually segregated the necromass for like root necromass and total necromass pool. So we segregated that to understand the contribution. Then run your final cell. And the summary file can be downloaded. On the left bar, you can see a summary subfolder under output files. You can download it from here. So it is downloaded. So if you want to make changes in the CSV file, input parameters, you can actually do that. What you have to do, like I'm changing here the BI value, 2.65, changing the BO value, 0.85.
Can you mention what those each of those are when you change them?
Oh,
or why
tracking
someone might
density of
change them?
organic matter and inorganic matter. I'm changing those values.
So if you go to the field, if you want to run this model in another site and you have field data, you can change these parameters to fit your particular site or sites.
Yeah, and probably I'm changing the root turnover rate, the KR value. I'm changing it to 0.65, and I will rename it. Save as browse. I'm renaming it as new. Site parameters, new. Oh, I have already one. I'm replacing it. So what you have to do, you have to drag that file, CSV file. I have the CSV file here. I will drag the file and put it in the input parameter folder. It will give some warning. You have to hit OK.
And that warning is saying that if you're using Google Colab, that nothing you do there is saved unless you connect it to your Google Drive. So make sure to download anything you want to keep. That applies to all of the
So you have
notebooks.
to change only the site parameters here in the cell 2. So rename the CSV. I renamed it with an underscore and new. I'm adding that. So I'm running it again. So it constructed the file path. It read the data frame after running the next cell. Then you have to hit run again for the other cells. So you just go down to see your graphs, new graphs. I changed only active brackish. So it couldn't fit inside the it couldn't fit inside the defined axis. That's okay, but still you can download from the output files to see what happened here. Active site, I guess this one. Yeah, it actually calculated all the value, but somehow it didn't show in the, probably due to the restriction of limits.
So can you explain the difference between this files for the individual sites versus the summary file? What is this file showing
So
us?
For the individual sites, it will have all 100 years value. So 0 to 99. So Python actually starts from 0 and 99. So it's 100 years value. So you can look at the cellulose refractory portions, lignin refractory portions for each of the cohort. And also you can track your cohort height. I mean, each of the cohorts volume from here, V in centimeter cube. You can also see the density, bulk density in different stages. Then your organic matter percentage, carbon density, you can see everything. So this is the file we changed, I mean, hypothetically. So this file, this will not give you a good value, obviously.
So one thing about the NUMAR model is, as Robert mentioned, it's meant to be run on large scales. So this is running for 100 years. It's not something you'd run for 1 year, and you can't look at seasonal changes. So it's looking at an annual average. And so this is what we're using to basically predict far into the future. Yep.
We are not changing. We are not incorporating any time variant sediment loading right now because it will be more complicated. So yeah, we have a plan to do that. We are talking to Dr. Wilson on the other day. So in the top portion, we are considering that in the top, there will be inorganic matter deposition. Somehow we are not considering any subsurface inorganic addition. So only inorganic pool in the subsurface or below ground we are considering as live root contribution, live roots ash content.
So each year the same amount of mineral inorganic is deposited on top. Is that
Yeah,
correct?
on the top.
But it's a rate that's determined today and applied every year for the next 100 years. Is it?
Sorry, can you ask him again? Oh, you were asking for site parameters?
I think it's SI. Is that the?
Oh, yeah, SI. SI is the
Can you
inorganic deposition.
can you pull up the slide where you have the table of parameters?
Sure.
Yeah. Yeah.
The plan currently, we're going to try to update at least every 10 years at some point for the sediment inorganic. We have to do a sensitivity analysis on how bathymetric changes change the hydrology and the sediment deposition rates. So currently, we're just shooting for an update every 10 years and see what happens to this model. But yeah, this is where we are now. It's here it is today, and this model takes over to project 100 years. But ultimately, it's going to have to be an iterative process.
Okay, so I guess the next thing
Yeah.
we can talk
Next
about.
one is landscape. So Alex will take over.
Okay, so this was for the sites, right? You can run this for an individual site, CRIM site if you choose. We're also building a landscape version. Okay, so basically instead of running on a single site, we run the NUMAR model for every pixel in the landscape from the Delta X domain. You can run this on every pixel or every eco-geomorphic cell, which I believe Robert presented earlier. For this example, we're running it on a very coarse pixel just for time's sake, but essentially you have the same input parameters assigned to each pixel. And so in this case, you could, again, if you had inorganic deposition rates across the domain, you could apply that differently each year. Yeah, so for each pixel, you run the NUMAR model. I'm gonna jump to the example. Okay, so this is 6B. Okay, so this one has several data sets that are required to run it. We have provided in the GitHub repository cropped and very coarsened versions to make them small, but you can also download them from multiple sources. Several of them are from Delta X, and I will talk about them as we go. So again, you can click the open and collab. Of course, it has an error. Awesome. That's really spectacular. Okay. Does anyone else get that error? Is anyone else trying? Okay. Was that this morning? Give me 1 second. Okay, did anybody actually download or running it on their local computer? Yeah? It worked. Okay. So it must be a collab issue. Okay. So yes, you can download it. Okay. Share. Right. Okay, so it's very similar to the last one, except that you need some extra things. So again, you're importing these packages, importing, this is the NUMAR model. So unlike the unit one, I didn't put the whole thing in here for you to see, but you can look at the source code and see how the model actually works. We also have some functions called input files, which will download the data. So this is actually designed to download through Earth Data Access. So everything on ORNL DAC is on the NASA website. And there are Python packages for querying the NASA data search and downloading it to your local computer. So if you choose to run this again, and you want to go through that whole process, if you just delete all of the files that I've provided, then it will download everything again for you and crop it and such. So this first cell is you're defining your area of interest. For this, I've included most of the delta X domain, but not all of it. So it includes Atchafalaya and part of Terrebonne. So you can change this. Obviously, you can't extend it past the delta X domain because then you won't have input data for it. But you can limit it to just Wax Lake Delta or just Ford Lake Bay, etc. Okay, and then we're going to download the input rasters. So if you want to go through this process, you need to have an Earth data login, and you need to import this Earth access package. And when you do that, it will ask for your username and password. Hopefully you all set that up because you had to get ORNL data. Once you do that, it will authenticate your login, and then anytime you try to download from Earth data, it will automatically provide those credentials. So the data sets we use for this, the first one is the AVERS-NG above ground biomass product. That's for spring and fall. Yes, so that's why we weren't going to do it in the collab. Yeah. So collab also has some memory limitations, not just storage and computation time, but if you try to merge large files, it will crash. We also use the ESA world cover land cover maps. That's mostly, so you can see land cover type. So this model is mostly designed for the marshes, correct, Pradipta?
Yeah. Marshes.
Yes. So right now we're running it on the forested areas, but theoretically you should probably limit it to only the marsh areas. We also use the CPRA vegetation map. This is mostly to get salinity zones. So we separate freshwater, brackish, and saline. I think as Pradipta and Robert mentioned, the different parameters will vary by salinity zone. And so we are going to be applying those model input parameters based off of the salinity type. You could also use the AVERS-NG vegetation class, but right now that product does not include brackish. It only has freshwater and saline, and then some other smaller classes like tall grasses. So the classes don't quite match up yet with the field data that LSU collected. Then we also use the output from the Delft 3D, I think it's called the broad scale sediment model. So this is not the model that Ali showed. It's one that is already on the ORNL DAC. It was run in the Atchafalaya and the Terrebonne basins. And you don't need to download the whole thing, but one of the products is called the inorganic mass accumulation rate, or IMAR. And so that is what we're using as the deposition on top of the marsh. So if you run this cell, since I've already provided you with the files you need, it should be very fast and it should really not look like much of anything. If you delete those files and re-download them, you'll see a lot of progress bars. So it's searching for the AVERS-NG data, for example. It found one granule and it's downloading it, etc. And then it's doing those for each of those pieces. So even the ESA land cover maps are available. You can just download them programmatically through Python. Okay, and this is again the parameters. So I think we saw a table earlier. So the SI, the inorganic matter deposition rate, is coming from the Delft 3D model. And the organic matter deposition rate is coming from a conversion from field data to go with what % of the deposition is. Yeah, you want to speak to that?
Yeah, so we had only data for organic and inorganic matter percentage. So we can use that ratio and convert the ASI value to OMS value. OMS means organic matter loading. So that was our process because we don't have much data for it right now.
Yeah, so the model provides the amount of deposition, and we assume the ratio of organic to inorganic is similar to what we found in the field, not they found in the field. And then the average NG above ground biomass data is used to derive the root biomass at the surface, and that was the equation that we saw earlier.
Yeah, so from above ground biomass, we can convert it to, I mean, using the ratio from Castaneda-Moa, we can convert that thing to root using root to shoot ratio and convert it for top 50 centimeters below ground biomass. And then we can apply our algorithm and get the RO value, I mean, the root biomass at the surface value. And the root attenuation rate is coming from directly the 6 sites we are working on right now.
And then the rest of the input parameters are basically like a paint by number. So the value for BO, BI, all the C's and F's and K's comes from the field results, and they're applied based off of active versus inactive basin and the salinity type. So essentially there are 6 classes. There's active saline, active fresh, active brackish, and then inactive saline, inactive fresh, inactive brackish. Each of those have a value derived from the field data, and those are applied to each pixel across the delta X landscape. So if we plot all of these things we've seen so far, here are the basins. So this is how we determine active versus inactive. As you can see, where you cut off the Chaffeli versus Terrebonne is a little bit different than the HUC that's published. The salinity type again is from CPRA. So we have saline. We've included brackish and intermediate together, and then the green is the fresh marsh. The average NG, this is above ground biomass. So again, that's used to determine the root biomass. And then finally, the mineral deposition from the DEL3D model. And then basically the next steps are setting up the CSV parameter file, it's formatted the same as we saw in the unit model, except that every pixel is a row, and then we'll loop through each row and run that through the unit model that we saw earlier. So to populate that CSV, we go through and add each parameter based off of their class. Again, that's active versus inactive and the salinity type. And that's all of these are the parameters that we need to populate. Then we calculate the organic matter. It's a function that it calls, but it's basically what he just described, how we go from inorganic deposition to organic matter deposition. And then again, we calculate the below ground biomass using a function based off of the root to shoot ratio, and converting from the root down to 50 centimeters to just the root biomass at the surface. Then finally, you have your run configuration or your input parameters. It tells you how many pixels you have. So in this case, we have 77,000. That's because we're running, I think, at 100-meter pixel size. So it's quite coarse, but probably still finer than what we will do for the final version. We save this as a CSV file, again, formatted exactly the same as the previous example. And then this is where we're actually running the model. So we're calling a function called run NUMAR, and that is feeding that CSV into the same code we saw earlier. And right now it's just telling you every time it does 10,000 pixels, it gives you a progress, so you know how long it's taking. I think in Google Colab, this version takes about 15 minutes. On your own computer, it might take less, hopefully not more. So if you were to increase the number of pixels, or if you wanted to run it for more than 100 years, that would make it take longer. And then finally, we get a data frame where, again, each row is a pixel in our delta X domain, and these are the different output parameters. So they're the same or similar to the ones that Pradipta showed, but I've also added total accretion for every 10 years so we can track accretion over time rather than just having one value at the end. And then finally, we can plot the mean accretion rate over our 100-year simulation. And that is this figure here. It's in centimeters per year. You can see the bright yellow is 3 centimeters per year. And you can also see the bright yellow, again, because the input parameters are applied based off of the basin and salinity type. You can definitely see the impact of those categories on the output. Now these values, I don't know if you want to speak to if these values seem reasonable.
Yeah, so this model is still in trial process, and I personally don't believe that 6 sites can represent the whole Terrebonne and active basins. And we need more data, to be honest. We need more data, and some of the data points were not very consistent, like inactive rate site. It was reported that in Feldspar, 30% is lignin. So if the lignin is high, we will get more accretion. So it retains in the soil. So we need more data. I mean, kind of comparable or feasible data points. And we need to taste this same stuff using more sites in specific salinity zones, like 4 or 5 sites at least, to testify this thing.
This also is strongly impacted by the model results. So if the Del3D sediment deposition model overestimates deposition, then this model is going to overestimate mean accretion rate over 100 years. Yes.
No, actually, we don't have any data for, 100 years sedimentation data or sedimentation map. So we need that before it.
Yes. So that's kind of what Mark was talking about earlier, where we're going to iterate on this process. So hopefully every 10 years, we'll run another Del3D or some model to get, and that will maybe have sea level rise and changes in mineral deposition. And then we'll run the NUMAR model for another 10 years and iterate on that process. So that the numerical model the hydrodynamic and morphodynamic model should be able to incorporate sea level rise. Yeah. Yeah. Yeah, so we have the salinity. That's how we're applying the input parameters now, is by salinity type. It doesn't actually incorporate elevation. We're assuming that comes in with the deposition portion. But yes, I think if we had more field data, you could also do a lot more correlation between these parameters and things that you can see from space. But I think with 6 data points, that's challenging. Yeah. The root biomass? I wasn't there, but I believe it was a 50 centimeter core and they did it in 10 centimeter, no, I don't know, 10 centimeter intervals. and then did the biomass. But then the model is taking only the root biomass at the surface. So they are using that decay function to get the surface root biomass. What year was that?
Yeah, we certainly have some data issues, I should say, and I should admit, because some of the datas we are working with right now are totally different from Cream's dataset. So there's like, you were talking about the biomass, below ground biomass. So from this model, we are actually predicting less biomass. So it is actually affecting, again, the root turnover rate. So it's kind of a circle. So right now, we are not sure what to do about it, especially for the landscape version. But we will definitely try with Cream's value and try to do interpolations and populate the ecogeometry morphic cell. Yeah, that is a better way, obviously.
Yeah, I think something that would be super useful is if there's above ground biomass with that below ground, because it's the ratios that we really need. So if we can see, or if we can estimate above ground biomass from Avarice NG or something, then right now we're using 6 ratios to get the below ground. But if we had more ratios by species or by vegetation type, then we could make that more spatially heterogeneous. Right, any other questions?
You can actually pull off side values, side parameters, unit model.
Yeah, you want
output.
for the
Organic carbon density, OC density, that one. So this organic carbon density, it is pretty consistent with what we are given with from the experimental data. And the problem is right now with the cell packing density of organic matter, inorganic matter. So in some of the data sets, we are getting low cell packing density of organic matter. In that case, it is predicting a little bit more soil accretion. So when it is making more soil accretion, so it is predicting a little bit more carbon sequestration. It is actually like, if you just see Melissa Bostian's paper 2021, you will see all the values are around 0.5 gram per cubic centimeter. So yeah. No, it's the output file for 4 marshes. So activate, yeah.
So these are 4 specific CRIM sites, and
Yeah.
they represent the different basins.
So active brackish is, if I'm not wrong, it's Creams 399. Active saline is Creams 322.
So those are both in 4 League Bay.
Inactive brackish is Creams 396.
That's Terrebonne.
Yeah, inactive saline is in where? Creams 421.
Yep.
So from this model, we're actually predicting lakes. So it is actually.
Do you want me to type in the CRIMS names? Too late. Do you want me to type in the CRIMS names for you?
So it's simulation. It's simulation.
For 100 years, yes.
Yeah, it's simulation. So from cesium activity testing, Boston estimated for active brackish, that is 0.76 or something centimeter per year. So 76 for 100 year.
That's the mean accretion rate?
Yeah,
Yeah.
mean accretion rate. And active saline. So for active saline, Andre took a core and did the stuff, and he got 0.5 centimeter per year. For inactive brackish, so he got 0.8 centimeter per year.
Do you have any validation of the carbon data, the carbon density?
So carbon validation is from actually Castaneda Moas, dataset. So he has published a dataset. It is available in the ORNL deck. You can download it from there and see their datasets.
Is this the paper that just came out? That one?
No, you can find it. I mean, he has already dataset in the ORNL deck in CSV files for all of the sites.
Oh, is this Edward?
Yeah.
Okay, sorry. Yes, Edward's paper.
Okay. All right. So yeah, thank you all for participating in our Delta X workshop. Most likely our last one, because this project ends in 10 months, 11 months, April next year anyway. So we're really hoping you guys can use all these models, improve them, use the data sets. So you're basically the continuity all these data sets. If you publish anything using these data sets, please go wild. Welcome any publications with all of this. You can tell us about your publication, so we add it to the website, so that we know there's already 2 publications by non-team members. Some team in Canada just downloaded the data and published it. So this is welcome, no problem. Have great PhD thesis